Our next speaker, Ed Boyden, is a professor of biological engineering and brain and cognitive sciences at the MIT Media Lab and the MIT McGovern Institute. He leads the synthetic neurobiology group and co-directs the MIT Center for Neurobiological Engineering, which aims to develop new tools and to accelerate neuroscience progress. Welcome, Ed Boyden.
Okay, let's see. Well, thanks for coming. It's really great to be here to tell you about what we're trying to do. Our technology development group is really organized around two fundamental premises. One is that the brain is particularly difficult to analyze and engineer because of its extreme spatial complexity. Brain cells are, of course, amongst the biggest cells in the human body. They can be centimeters in spatial extent. And yet, if you care about neural connections, if you care about proteins, if you care about gene products, those are nanoscale and quite often organized with nanoscale precision. Neural connections and so forth are very, very tiny. So how on earth do you cross those spatial scales if you really want to get to what you might call ground truth engineering and analysis of neural circuits, to really engineer computations to do what you really, really want them to do? The other issue, of course, is time. Neurons, of course, have amongst the highest speed. Whoops, can we have the sound off? They don't need the voiceover because I'm here. Okay, great. Well, now you've heard the whole story, so I can sit down. Okay. Yeah, just if the audio can be turned off. That's for when I'm not here. Great, thank you. Okay. So yeah, there are very brief electrical pulses that last perhaps a millisecond in duration, and neurons communicate using chemical exchanges that are also very brief. So I often say one of our goals in our group is to develop tools to help us cross space and time if we want to really understand and fix the brain. So jumping right in, we've been working on lots of tools that help us do that kind of crossing. And I'll tell you today about a couple of these different technologies, one on the space side and a couple on the time side. And I hope you see here this sort of interesting directionality of how we try to approach problems. We pick a big problem like space or time. We try to develop tools that let us confront it, and then we try to seek extremely practical, deployable technologies that we can bring out into the world to help with diagnosis and even treatment of disease. So the first thing I want to talk about today is a technology that our group has been working on that we call expansion microscopy. Now, what's that? Well, if you take a piece of brain tissue or a brain circuit and try to zoom in using electron beams or super resolution methods, it's really hard to take extended three-dimensional pictures that are accurate all the way down to the nanoscale. So we thought, what if we tried to do the opposite and physically blow the brain up to make it bigger? So this works on preserved brain tissues, but even those, we think, if you could expand them and map them, maybe you could use this technology to really try to look at the wiring of neural circuits with great precision. Now, how does this work? Well, we want to install polymer chains, and really it's just the same kind of polymer that you find in baby diapers, so densely throughout a piece of preserved brain tissue that they'll wind their way around and between all the biomolecules. And so to do that, we combine two ideas. One is a very old idea from the MIT professor Toyoji Tanaka, who unfortunately passed away relatively young from a heart attack, but he had, was one of the pioneers of studying these responsive polymers that undergo huge changes in size or shape, these phase transition-like effects when you expose them to the right kind of environment. So if you take baby diaper polymer and you add water, it'll swell enormously, absorbing the water, and it'll become 1,000 times bigger in volume. The other thing is, well, how do you actually install these polymers in tissues? And again, if you go back to the 1980s, groups like Christine Dreyer's group at the Max Planck Institute were taking specimens, soaking them in these polymer building blocks or monomers, and then synthesizing them into these spiderweb-like networks. So we thought, let's combine these two ideas, one from phase transition physics and one from histology, and see if we can make a new technology that would allow us to pull apart the building blocks of life until they're far enough apart that you could turn a cell like the one on the left to one like the one on the right and have like a constellation of biomolecules hovering in space, anchored to the polymer. If you could do that, you could actually map the building blocks of the brain, maybe with enough resolution that you could someday simulate it in a computer. Maybe you could develop more interesting kinds of AIs, perhaps. But we'd also maybe learn a lot about how neural systems compute. So how does this work? Well, we had to invent three kinds of chemistries. The first was an anchoring chemistry. So in this cartoon, biomolecules are shown in brown, and little anchors shown in purple are diffusing to the biomolecules and connect. So now we have a handle. We can apply force to the biomolecules and pull them apart. The second idea is you have to form those polymer meshes. And again, going back to the Dreyer paper from several decades ago, people have been embedding specimens in very, very similar molecules to what we've been doing, although not the same kind that undergo the phase transition effects and vast changes in volume. So when the polymers connect to the handles, we now have the force generator, the polymer swelling, and the force conductor, the purple handle. Finally, we have to loosen up the molecules. They don't like being pulled apart. They're very happy where they are. We use detergents or heat or even enzymes to do that. And then finally, we add water. The polymers absorb the water and expand apart from each other, but now the building blocks of the brain, biomolecules, proteins, pieces of DNA, pieces of RNA, come along for the ride. So we published this idea a couple years ago. You can see here an actual specimen in panel B of mouse brain tissue in this case. And in panel C is the same piece of mouse brain tissue blown up by about 100-fold some number of hours later. And on the left-hand side is a schematic of what we think is happening at the polymer level. A dense spider web absorbs the water and becomes a less dense spider web. So the beauty of this now is you can blow things up and take images that are nano-precise in a scalable way, very often using ordinary optics. In fact, my colleague at Harvard, Ali Karabassani, has a paper where he used expansion to blow up specimens of bacteria and then to use basically modified webcams, not unlike the kind that you might find on phones or on laptops, and he could actually image bacteria. So imagine an era where we can do diagnosis for almost zero cost. In neuroscience, one of the key things we can do now is we can actually image large-scale neural circuits with the precision that you need in order to identify individual pathways and connections. So this is a piece of mouse hippocampus, a region involved with, amongst other things, memory formation. And we're using a technology called Rainbow, developed at Harvard, to color code neurons. You basically put different colors in different cells. And there's a little white square there. If you look at the top, just above it, there's a zoom in. It's very blurry. That's before expansion. But the upper right is the same specimen as the middle top, but after expansion. And now you can clearly see pathways that were invisible before expansion because they're below the resolving power of microscopes. Or here's another example where we can actually take a sample and blow it up multiple times. So form the baby diaper-like polymer, blow it up, form a second polymer in the space opened up by the first, and blow it up again. And what you can see here in A is a non-blown up brain, really hard to see. B is after 1 round, and C is after 2 rounds. And if you look carefully, these are speckly images. So we think we're actually getting down to single molecules here. And so the idea that maybe you could map out the location and identity of all sorts of biomolecules throughout the wiring of a neural network to the resolution and completeness to do a simulation might not seem so science fiction anymore. Now, what often happens when we build a technology to map brain circuits or to control brain circuits is that we find other uses. And one of the things that we've been doing is applying this to cancer diagnosis. So we did a collaboration with several other groups at Harvard Medical School, and we developed a version of this expansion protocol that works really well with human tissues, just tweaking the recipe a slight amount, because human tissues are prepared in a certain way in pathology. And what you see here are a bunch of before versus after images, in this case from human breast cancer patients. In panel L versus panel M, what we have here is actually something slightly different. These are HER2 negative and HER2 positive breast cancer specimens. And by expanding them, one of our hopes is that we can decrowd molecules to the point where we can count them more accurately. Or by expanding them, we can get more detail. Here are a bunch of different cancers. I know it's a bit of a crowded slide, but if you just focus on a few of these panels, for example, these two right here, that's before expansion, that's after expansion. After expansion, you get a lot more detail than is possible beforehand. That allows us to see features. Oh, we can use laser pointers, technology. You can see features after expansion that you can't see before expansion. And so to put this to the test, we collaborated with Andy Beck's group, who does a lot of machine learning for trying to make automated pathology strategies, and when he was at Harvard. And we decided to tackle a problem, which is early breast cancer diagnosis. So it turns out that many early breast lesions in human patients are very hard to diagnose. The disagreement between pathologists can be as high as 50%, a Journal of the American Medical Association editorial recently pointed out. So what we did was we, whoops, sorry, try to use the laser pointer now. We can take a specimen, and then we can expand it. And then what Andy did was to train a machine learning classifier where the training signal was a committee of pathologists who would vote on whether a specimen was one kind of precancerous lesion or a normal sample or a full-blown cancerous lesion. And after expansion, the quality of the machine learning was much higher than before expansion. And so one hope is that maybe this could be very helpful for early diagnosis of a variety of diseases. In fact, we just recently spun out a company to try to take this through clinical trials for a variety of cancers, kidney diseases, and other diseases which are very subtle. Expansion is spreading pretty rapidly. There must be a paper coming out every week or so now where people are applying it to expanding entire mouse brains, looking at circuits involved with addiction, bacteria I mentioned earlier, changes in proteins, human brains, flies, and cell division, and everything in between. So in summary, this first part of the talk, we think we have a way now to map extended three-dimensional objects with the spatial precision of molecules and cellular compartments. And it's really simple to use. All of the protocols are on our group website, and we host visitors nearly every week here at MIT to learn how to do it. But this, of course, can only work on preserved tissues. You can't expand a living thing, at least not that we can figure out. And so it leaves open this question of time. So I want to spend the last half of the talk about technologies that we think can help us analyze high temporal precision functions in the brain, and also maybe to engineer the brain in fashions that could yield new insights into how to treat diseases like Alzheimer's and other kinds of disorders. So there are lots of ways to control electrical activity in the brain. So one of the nice approximations that works pretty well in the brain is that neurons are computing with electrical signals. If we can control those electrical signals, then we can have a great deal of ability to regulate brain functions, computations, and pathologies. And many, of course, attempts, electrical stimulation, drugs, some of the things you've been hearing about at the conference, have been brought forth over the years. But one of the big questions is, could we actually build technologies that approximate the spatial and temporal language of the natural brain? Can we get down to single cells and single electrical pulses in the brain? So many years ago, when we were both students, my co-inventor, Carl Deisseroth, and I started brainstorming. And we just went through all the laws of physics. What can we do with ultrasound? What can we do with magnetism? What can we do with mechanical perturbation? And finally, we decided that if we could aim light at individual cells, then you could control them as precisely, both spatially and temporally, as maybe you could ever do. And of course, you need to get light into the brain. The brain, the first order, doesn't feel pain, so you can bring in optical probes. There are hundreds of thousands of people with neural implants in their nervous system for stimulation with electricity, and so maybe one day we'll see something like that for optical probes. The big question, of course, is how do you make the neurons controllable by light? And so this is where we were just very lucky. There are lots of molecules in the natural world that have been studied for nearly half a century that convert light into electrical energy or other electrical signals. So if you've ever seen really salty water with an orangish or reddish hue, there's a good chance that some of the microbes there had a pigment, and that pigment, there's a good chance, was a light-driven ion pump, a 7-transmembrane protein that sits on the boundary of the cell, and when hit by sunlight will cause an ion, in this case a proton, to move from one side of the molecule and the membrane to the other. So it's a pump. It stores energy, converts light into a chemical gradient, a primitive form of photosynthesis, if you will. So these were discovered in the late 1960s, early 1970s by Oesterheld and Slakenius, and they named this bacterial rhodopsin. About a decade later, people found halorhodopsins, light-driven chloride pumps from the same microbes. These are very similar, and also except for a few key differences. And then around the turn of the millennium, people found light-driven ion channels in algae, and they named them channelrhodopsins. And these, of course, being channels, don't store energy, but you can still use them to signal light to the organism. And in this case, single-celled algae use this to swim around and navigate in ponds or other bodies of water. So we call this now optogenetics, opto for light, and genetics because we lucked out and there was a gene just lying there in plain sight in a whole class of microbes. And what we found, and our colleagues and collaborators over the years, is that there are members of each of these three classes, the proton pumps, the chloride pumps, and the ion channels, that you can find that are safe, effective, and fast enough to work in living, fragile neurons. So if you pump protons out of cells, you can shut down the electrical pulses. You can delete the cell temporarily, which is very helpful. You can shut down, for example, people have shown you can shut down an epileptic seizure in a mouse model. People have shown that you can blockade a part of the brain and figure out what it's necessary for. I'll show you some examples in a second. If you pump chloride into neurons, then you also shut them down. And if you bring positive ions into cells through light-driven ion channels, then you can activate them. So let's give you just a couple examples. This is work from Akihiro Yamanaka's group. They made a transgenic mouse that expresses a halorhodopsin, the silencer, in some of the cells that are atrophied or gone in patients with narcolepsy. And what they found was that they would shine yellow light from a laser down an optical fiber at these cells. And what would happen is these mice would fall asleep. So you can use this technology to try to parse out what given cell types do and function, both in normal as well as in different kinds of states of the brain. And what we do in our group is to try to extend these technologies to their physical limits. So a couple years ago, we revealed a molecule that we called JAWS, which is a silencer which is drivable by red light. So red light you can shine across even the skull of an intact, freely moving mouse in this case, and you can shut down neural activity many millimeters deep in the brain. And recently, my colleague Bob Desimone completed a preclinical study with non-human primates showing that with a single optical probe, you could shut down many, many, many cubic millimeters of cortical circuitry and actually cause the first optogenetic silencing induced changes in primate behavior. So that's a very helpful data point in this quest. What about activation? This is some work from the Fiorello lab. They use this alphabet soup of viruses and transgenic animals to make dopamine neurons sensitive to light. And I think many of us have heard of dopamine neurons, or sometimes referred to as the pleasure center of the brain in the popular press. But an open question is, well, if you activate them, what do they really cause? And what kinds of normal as well as abnormal changes, learning, but also maybe addiction or other kinds of changes will occur? So Chris made these mice and then put an optical fiber into the brain, coupled this time to a blue laser. And every time the mice would go to a certain point in a little box, they would get a pulse of light into their cells. Every time the mice go to a different point, nothing would happen. And here's the movie. Mice go, poke their nose into a little sensor, get a pulse of light. They do it again, get another pulse of light. I think you're seeing the pattern here. The animals are basically working for light. And this is sort of a proof that you can activate these sets of cells very briefly. And that's enough, at least in the context of this task, to make the brain do more of what it was just doing. One of my favorite recent examples was was led by my colleague here at MIT, Li-Wei Tsai. And she's an expert on, amongst other things, Alzheimer's disease. And so their group used the blue light activator, the same one I just showed you, to drive neurons in the brain at 40 times a second. And these are actually Alzheimer's model mice. And amazingly, the amyloid plaques and other changes that are molecular hallmarks of Alzheimer's disease, many of these got cleaned up, which is kind of surprising. We don't usually think of neural activity as a therapeutic for neurodegeneration, but they replicated this on 3 different mouse model lines, and it was a very exciting finding. And then Emery Brown, also a collaborator in this project, was brainstorming, and the idea arose to have animals that are not optogenetically engineered, normal Alzheimer's model mice, if you will, have them just watch a blinking light. And what happened was the blinking lights would actually trigger responses in the retina, which would entrain the brain, and that was also enough to help clean up some of the amyloid plaques and other molecular hallmarks of Alzheimer's. So imagine an era where movies could be developed that might actually treat a neurodegenerative disease. And Li-Wei and I now have co-founded a company to do the human trials with the hope that we can develop extremely non-invasive strategies for helping treat one of the most intractable brain diseases out there. So again, in our group, we're trying to really extend these technologies to their physical limits. I showed you the stuff earlier was driven by blue light. We're back to optogenetics now. No longer just movies. We have brought the optogenetics back. We found molecules respond to red light. Red light could go deep into the body, so we can activate neurons with red or even infrared light, and that hopefully will allow us to activate very large volumes of neural tissue. We work on a lot of other tools as well, and we're almost out of time. I think we have only about 2 minutes left. But I just wanted to talk about how we've also been trying to form collaborations that allow many kinds of neuro technology to emerge from looking at how to connect the dots between existing engineering fields. There are many fields of engineering that have not yet been applied to the brain. And so one of the great opportunities here is to build tool sets that allow us to really get down to the ground truth understanding of how to analyze and repair pathology, but then to develop really practical tools that can be used ideally with human subjects in order to help make life better. And so I've given you 2 examples now, one where we're mapping the brain for ideally the quest to understand the computations, but might lead to cancer diagnostics, and then one where we wanted to control neurons to figure out what they're sufficient to trigger, which might lead to inspire new sensory modality stimulations for Alzheimer's disease. But we're working on automating surgery. Are there strategies to develop robotics that can actually interface to the brain in an automated way? Robotics have been very revolutionary in vitro sciences like genome sequencing. Why can't we do the same for the living brain? We've been working on ways to try to use laws of physics to develop new non-invasive strategies for interfacing to the brain. Can we treat the brain like a radio and play back multiple frequencies so that only where the 2 frequencies collide do you activate deep structures in the brain? Can we build wireless devices, all sorts of other devices that bring together different technology trends in order to build strategies for interfacing to the brain? And then finally, nanotechnology. Can we build nanotechnologies that allow us to really scale these kinds of neural interfaces that we've been talking about so we can cover the 3-dimensionality of the brain without losing sight of the precision that we need to confront if we really want to interface the brain at its natural level of operation. So I think I'll end there, and thank you so much. All right.
I ask you a question.
Yeah, sure.
So, what is my conclusion using the non-contact optical methods to manipulate cells to pre-functionality for therapeutic purposes?
Yeah, I really view two big goals. One is to combine observing the brain, controlling the brain, and mapping the structure of the brain into a holistic set of data so we can build true computational models of neural function. Can you model a thought or a feeling or a sensation? The second is, by using these three kinds of mapping tools, can we actually pinpoint the proximal causal biomarkers of disease that would allow us to pinpoint how to treat them better? And then a third emerging strategy that some of the non-invasive methods that I mentioned at the very end is, can we develop very practical strategies for controlling neural circuits in awake behaving subjects without surgery so we can help as many people as we can? I would say those are the three main goals. Yes?
So in your talk, actually, previous talk you heard about.
Might you be able to use the microphone?
Sure. I can talk louder. So in your talk and the previous talk, we heard about the number of neurons corresponding to image or the temporal resolution or the actual spatial resolution. So what is the actual number you think we should shoot for? Because not anytime soon we're going to image the whole brain in a very, very high resolution. So why a million? Why a specific spatial resolution? So what is the target?
Well, my hope is that we can actually interface to the whole brain. I mean, if you look, there's some great papers by Michael Brecht or Yang Dan where they take a mouse or a rat and they stimulate just one cell. And that's enough to change the entire animal's behavior or the entire state of the whole brain. So our personal mission anyway in the group is to try to scale up as much as we can. Because we don't know where the obvious limit is how do you know for sure that any given number is all we ever need? So the good news is that we can always use less things later, right? If we built a 5 million channel device and we only need 1 million, we can always turn 4 million off. That's a lot easier than trying to make 4 million more. So, but yeah, it's I use this phrase ground truth a lot. How do we know when we know something? And I think part of the problem in neuro is we often don't know when we don't know something. So we have to, I think, assume our ignorance and try to build tools that are powerful enough to give us, in the worst case, the answer. But in the best case, we get more than what we hope for, which is always nice. Yes?
Diagnostics for breast cancer in terms of sensitivity, specificity, how accurate it is.
That's absolutely right. So what I plotted there actually was the area under the curve of a receiver operating characteristics. That what actually is the exact curve you're talking about. So 0.5 area is chance and 1 is perfect. Before expansion, the numbers were 0.65 to 0.85, let's say. And after expansion, they're at 0.9 to 0.95. But this was a very small study, only a couple dozen subjects. So right now, the goal is to do a large-scale study. So I wouldn't take the exact numbers too seriously, given how small the study was. Yes?
What about the 40 hertz? Something magical about 40 hertz?
That is a great question. Yeah. Oh, what was the question? Is there something magical about 40 hertz? Yeah, I think the answer is maybe. 40 hertz oscillations have been found in many cases where people are paying attention or linking prefrontal areas and early sensory areas in ways that maybe optimize communication in certain ways is one theory. In patients with schizophrenia, people have reported lower amounts of 40 hertz oscillations. And also there are very specific cell types that seem to be, when you activate them, they drive this 40 hertz resonance. So lots of clues from different parts of the field, clinical, neuromodulatory, and so forth, that point out a certain kind of cell and a certain kind of resonance as being important for interesting cognitive properties.
So rephrase my question. On this mice, did you try other frequencies and it did not work and only in 40 did it work?
That's right, yeah. So in the project that Liwei led, if you drive it at 10 hertz or 20 hertz or 80 hertz, then you don't get the effect. Yeah, 40 is indeed somewhat special. All right, well, thank you. Appreciate it.